Hello, this is your review for the Unit 0 test. It's just going to be a quick verbal review where I talk about each term. As a reminder, if you fill out your review sheet and turn it in right before our test on Tuesday, you'll get an extra 5 points on your test. So let's get started. So the perspectives, the biological perspective versus evolutionary, both do involve genetics and understanding how the brain has impacted the body. The difference is the key thing with the biological perspective is it's really just looking at current things going on in the body and through genetics and DNA and how those are affecting a person's behavior. The evolutionary perspective is much broader, and it's about traits that a person may have that allow them to adapt to the environment, that help boost their chances of survival, help increase their reproductive success. And when we're talking about traits that are genetic with the evolutionary perspective, we're generally talking about things that have been passed down from many generations that have adapted and are advantageous for survival. Number 2, case study versus naturalistic observation. Both are non-experimental methods in gathering data and observing in a research scenario. In a case study, it's usually a narrow group of subjects, usually something rare or unique. Our example that we used in class was serial killers, but it might also be something like a rare cancer type. And in it, they're gathering a large amount of data, but it's diverse data. It could be brain scans, it could be personality tests and intelligence tests and personal interviews. So lots of different information. So they'll get a lot of information, but the big challenge with case studies is they are time consuming, they're expensive, and you cannot generalize them to the overall population. Naturalistic observations, we know, is watching people or animals in their natural setting in which they are unaware that they are being watched. We relate naturalistic observations to the Hawthorne effect, which is the idea that if people or animals know that they're being watched, it causes a change in their behavior. For number 3, we've got some different types of psychologists. A clinical psychologist earns a PhD in their field, and they're going to treat behaviors, particularly mental illnesses, with a non-medical approach. Usually it's going to be talk and behavioral therapy. A psychiatrist is also going to treat people with mental illnesses, but they go through medical school, so they're a medical doctor or an MD, and they can take a medical approach. And an industrial psychologist does not treat patients, but rather applies psych principles to the workplace. Number 4, a confounding variable is any extraneous variable in addition to the independent variable that has the potential to interfere in an experiment. So if you can catch it ahead of time, an extraneous variable becomes a controlled variable, but if not, it's confounding. Our example from class in the Axe body spray experiment was where Johnny posted racist tweets on social media, and then after they conducted the experiment, they couldn't tell if their dependent variable was affected by Axe body spray or by his comments on social media. So confounding variables are generally not a good thing. They may not totally ruin an experiment, but they definitely would have to be reported and can affect results. Control group versus experimental group. If you see these in a description, that's a clear indicator that that type of research is an experiment. The control group is the one that is not getting any special treatment. So they are controlled in the sense that we're not doing anything to them. The experimental group is the one that gets the independent variable. So if we're talking about a medication, the experimental group would get the new drug. The control group would get a placebo. If we're talking about Axe body spray, experimental group was Johnny wearing Axe body spray. Control group would have been him wearing no body spray. So the experimental groups get special treatment. A correlation coefficient is represented by the variable R, and that is how we measure the strength of a relationship between two variables. It would be represented between the numbers 0 to -1 for a negative correlation and 0 to +1 for a positive correlation. You do want to go back and look at your notes on correlation coefficients and make sure you can understand that number line is that any number close to 0 would indicate that there's low or no relationship between variables, and a number closer to 1 or -1 would indicate a strong relationship. Number 7, debriefing relates to ethics in psychology, and it's the idea that following any kind of research, the participants need to be debriefed or told what was happening in that study. This is specifically important if there's any type of deception, even if it's not something that would have been harmful. The participants have the right to know what was being studied. That's debriefing. Empiricism, sometimes we use the form empirical or empirical research, is the idea that research should be based on observations, not just random guessing, not theoretical approaches that have no way to be proved to be valid. So empiricism says that research has to be based on some type of observations. An experiment versus a correlational study. So in an experiment, we have an independent variable and a dependent variable, and really a correlational study can have that as well. But the key thing here is that in an experiment, we're going to use an experimental group and a control group. We're going to use random assignment is another key phrase. We're getting samples from the population, and we are following the rules of ethics. A correlational study can be very similar, but often these are done when you cannot ethically conduct an experiment. So for example, if you wanted to see the effects of drugs on unborn babies, you cannot ethically conduct an experiment like that because you would have to give women who are pregnant drugs, and that's not ever going to get passed. So instead, you could do a correlational study where you analyze or you compare the relationship between women who did happen to do drugs while pregnant to women who did not. And in that case, we can compare results, but we cannot 100% determine cause. So the experiment, the big thing is that we can determine cause and effect. The correlational study, we cannot. Falsifiable versus generability. These are not really a one against the other, but they're just both terms here. Falsifiable is the idea that a hypothesis can be proven wrong. So the idea, if we have a medication and we're saying that this medication decreases depression levels, then our hypothesis would be if this person takes this medicine, then their depression would go down. So that is falsifiable because it is possible that after conducting their experiment, there may be no effect on their depression. So that means it's falsifiable. Generalizability means that research that's done in a study can be used or generalized across the population. So let's use that same experiment that if we tested out that drug and we wanted to say this drug works and it can be used for anybody else who is similar, who has anxiety, then that study had to have been valid in that it used random selection and random assignment. Because if you're using that randomization, you're ensuring that you have a broad enough or diverse and fair representative sample. Remember that representative samples can be done by matching the population characteristics or by gathering a random sample. The next one, number 11, hindsight bias, is a flaw in research where a person tends to think that they would have been able to predict data all along after the results have shown up. Humanism is the perspective that focuses on people's ability to make choices, to, once their needs are met, to approach self-actualization and reach their full potential. It was developed in contrast to psychoanalytic and the behavioral perspectives, which made it seem like people were simply the products of their environment and their unconscious mind. The independent versus dependent variables in an experiment, the independent variable is the one that we manipulate, and the dependent variable is the one that we're measuring. You'll want to be able to recognize in scenarios what the independent variable is and which is the dependent variable. Informed consent versus informed assent, both involve getting permission to participate in a study. Informed consent is getting permission from participants in which you're telling them all the potential dangers that could be involved. You are allowed to deceive them about what might actually be happening in the study, but not about something that would keep them from wanting to be in the study. And then informed assent involves minors. So when minors are not able to give their own consent, this is looking and trying to gauge by body language and actually asking them questions if they're comfortable with being in the research as well. large and or high versus small and low standard deviation. So we'll start with low. So low standard deviation means that data is close to the mean or the average. Our example of this were the speeds driven on the two streets. We compared one to the front of the school and one compared to the back of the school. So I think it was like Perfection Boulevard or something like that. The speeds on the front of the school, we said, would have low standard deviation. And that would be a distribution curve that is narrow and kind of high. And that's because most of the scores are close together near the average. We said that Wild Street would be like the street behind the high school, which has high standard deviation, which would be a really wide curve. And that's because the scores go way low and way high from the average. Measures of central tendency are statistics that we use in psych once we have gathered some kind of numerical data. This is what we did in our cookie lab. The mean is the numerical average, where you're adding numbers together and dividing by the quantity of numbers. And that's also the number that we would put in the middle of a normal distribution curve. The median is the data number that's in the middle of all the others. To do that, to calculate that, you'd have to put the numbers in numerical order and find which number's in the center. If there are two numbers in the center, you're going to add them together and divide by 2. Mode is where you're finding which piece of data appears the most. And again, it's possible to be bimodal or maybe even trimodal, where you have two numbers that appear the most number of times. And then range is calculated by subtracting the smallest number from the largest number in your data set. A meta-analysis versus a survey. We'll do survey first. A survey, we know, is simply a questionnaire, but it's open to lots of different types of bias. A meta-analysis is a combination of lots of similar studies to find common patterns and just really verify data. So it could be a bunch of surveys that are similar, like if multiple high schools had conducted surveys about if their students are going to go to college, then a meta-analysis could be conducted that analyzes all of those. It could also be similar experiments that were done together or naturalistic observations that were done in maybe various locations, and then comparing all of their responses, and then that would be a meta-analysis. Nature versus nurture. This is the idea that nature, which would be genetics and DNA, influence behavior, versus nurture, which is environmental factors. A negative skew versus a positive skew. You'll want to go back and look in your notes at this, but both of them, the way you can remember it is where the curve is being pulled towards. If it's being pulled towards like the negative side of a number line, that would be a negative skew. And when it's being pulled towards the positive end of the number line, that would be a positive skew. Operational definitions are when we are clearly explaining both our independent and dependent variables. We are quantifying and clarifying what exactly we mean so that when somebody else tries to conduct the same experiment or study, it could be a correlational study or really any type of study, but really going into detail, that they'll be able to conduct it the same way. For example, if we were doing a study on an anxiety medication and how well it reduces anxiety, our operational definition of the independent variable might be that they will take 10 milligrams of the anxiety drug twice a day for 2 months. And then their definition would be how they measure the anxiety, and that would be the person rates their anxiety on a scale of 1 to 10 3 times a day for the whole 2 months. So we're very clearly identifying how we're going to give the independent variable and how we're going to measure the dependent variable. The placebo effect versus sampling bias. These don't really totally go together, but I just put them here in the same one. The placebo effect is when a person is taking a placebo, which is something that's given to a control group but has no medical treatment, yet they still have some sort of change in their behavior. So I'll use that anxiety medication example again. So let's say that our control group, they get the placebo and they report that they feel better most of their days. If the independent group, or sorry, the experimental group also reports pretty much the same response, that's going to tell us that our independent variable, the medication, really was not super effective because there was not a big change in what the placebo group responded and how the actual medication group did. So it's normal for there to be a response where people do start to feel better, and that's the placebo effect. That just because they know they're getting some treatment, they're going to feel better, even if they were only getting a placebo. So in an experiment with a drug, we need to see a significant difference between that experimental group's behavior versus the control groups. So we know it was not just a placebo effect, that it was actually our independent variable, the medication, that was causing a change in behavior. Sampling bias is when there's some error in who we gather to be in our study, whether it's a survey or an experiment. Sampling bias can be broad. It could be something like only including wealthy schools in surveys about who's going to college. We've talked about convenience bias. That was in our assignment, and that's a type of sampling bias where it usually goes with location, where you're only gathering people who are close to you, like deciding, well, I'm only going to do this at Red Oak High School and Waxahachie High School because those are close to me. Whereas a better sample would be every high school in Ellis County. Population versus sample. Population is the overall potential group that you could study. So again, if we're studying high school students and the percentages that would want to go to college, our population could potentially be every high school student in the state of Texas. But realistically, we cannot study that many. So instead, well, we're going to take a sample of that, which would be every high school in Ellis County. A positive versus a negative correlation. Positive is when we have two variables that relate and that they both increase together or they both decrease together. So for example, the farther you drive, the more gas you're going to use. The less far you drive, the less gas that you use. Number 24, psychoanalytic perspective versus cognitive. Both involve mental processes, but the psychoanalytic perspective, that was Freud's and primarily involved the unconscious mind. Cognitive perspective involves mental processes, including thinking and problem solving and how we learn language. So it is still mental processes, but it's really from a more empirical standpoint today in that we can conduct interviews, we can do intelligence testing, to try to figure out what people are thinking, whereas there's really no way to show what would be going on in somebody's unconscious mind. Random selection versus random assignment, two very confusing terms just because they both have that random in there. So the selection has to do with how you're getting people from the overall population. So when you select who will be in your study, it is random. So we talked about using maybe a computer to randomly choose 100 students from Red Oak High School. So that would be random selection versus going and getting every kid whose last name starts with the letter A. That would not be random. We picked all the kids with the same last letter name, first letter. So that's random selection. When we get them randomly, it can be drawing names out of a hat or maybe random computerly generated list. Random assignment says once we have that randomly selected group, that we are going to randomly assign them to our experimental or our control group in an experiment. When we do both of those, that increases the generalizability of our study, that we can say our results match a broader population, and it also makes sure that our study is valid. Regression toward the mean says that when we have data, like for example, intelligence test scores, the first time that you give that to maybe every student at Red Oak High School, you may have some random outliers that are really high or really low. But if you repeatedly gave them that test multiple times throughout the years, you would see that there's those outliers gradually regress toward the mean. You may still have some outliers, but in generally the scores come out closer to the mean. A representative sample versus a random sample, both allow for a study to be generalizable, but the difference is how you get your participants. With a representative sample, you are intentionally matching your participants to the overall population. So for example, if we were looking at low income, middle income, and high income families, and we knew that in a town, that in our area, that we wanted to include 33% of each income level in the study, that could be representative sample. A random sample would be randomly choosing, you put all the names of the people's families in a hat and you pick out 100 names or whatever. But it's random. Both would create valid samples. Sigmund Freud, he is the founder of the psychoanalytic perspective, one of the few names that will actually show up on the AP exam anymore. But psychoanalytic perspective, example of unempirical research. Single blind versus double blind experiments, these are both done in order to eliminate participant bias and researcher bias. With single blind, the participants in the study don't know if they're part of the experimental or the control group. They don't know if they're getting real medication or a placebo. So that way, when they're reporting their feelings to the researcher, they're not just saying, oh, I feel like I'm getting better, but it's because I'm taking the medicine. They don't actually know whether or not they are. Double blind, the researcher does not know either who are the experimental and control groups, and that prevents the researcher from having their own bias while analyzing the participants. Social desirability bias versus response set. These are both problems that could appear on a survey. Social desirability bias is a type of self-report bias in which the person responds in ways that make themselves sound better, that maybe they say, I have never lied, or I'm more honest. A response set is when a person responds the same way to every single question. So if we have like agree, disagree, strongly disagree, and if everything they put is strongly disagree, it appears that the person didn't read all the questions, that they just went down the row and bubbled in, and that is a response set. Sociocultural versus behaviorist perspective. These are, I paired these together because they are similar in that both have the influence of the environment on their behavior. Behaviorist says that you were you were taught by your environment, either through observations or by classical or operant conditioning, or somehow you were taught through reinforcement or punishment. Sociocultural perspective says you were also taught by your environment, but it was a cultural aspect of your environment. So it's a more specific type of an environmental thing. For example, you were taught through your religion. You were taught because of your gender or because of your age group or socioeconomic status. Standard deviation and the normal distribution curve. The standard deviation, remember, measures the distance from the mean. So in a normal distribution curve, I'm actually going to do this one in a separate video, so I'm going to pause and save that one for later. Statistically significant tells us that there is a less, and this is measured with a p-score, so p for probability, that the probability is less than 5% that our data was due to chance. But instead, the independent variable is what influenced the dependent variable. So our data is good and then it's not due to chance. All right, I will post a separate video reviewing standard deviation and normal distribution curve.